Great, thanks Ryan. Thanks for coming today. And I guess you couldn't go to Florida or somewhere warm. So I'm glad you're here. Welcome to the two people that are going to watch this video later.
200.
200, right on. Okay, so we're going to talk today about some pathogens and potato early dying. I'm sure you're all familiar with potato early dying, so we're going to talk about some updates. This is going to be very informal, sorry. Please ask questions as we go along, okay? And I just want to acknowledge Dow Chen, a researcher at Agriculture Canada in Fredericton as well. He's the Agriculture Canada lead on the CanPEDNet project. The reason why we did this project, and it started basically the fall of 2018, is that we acknowledge that the yields in Atlantic Canada could be higher. The next was that Manitoba and Alberta have a deficit in potatoes. We don't grow enough potatoes to serve particularly our processing industry. So we needed to increase yields, and particularly our older land was starting to decline in yields. Okay, so we wanted to see about how maybe we can resuscitate some of that land. And just in general, potato early dying is acknowledged as a severe issue for yields in most potato growing regions in temperate world, particularly in North America. Okay? So it's an issue here, it's an issue in New Brunswick, it's an issue in Ontario, it's an issue in Manitoba, it's an issue in Alberta, it's an issue everywhere. Okay? So it's one of these topics that's really nice for a network and a national project. A lot of researchers in all those provinces that I mentioned, and then also a lot of grower groups, including yours, contributing cash from your checkoffs to the project and activities, and then even lots of other industry partners that are non-growing groups, so processors, for example, and some chem companies. This was under the umbrella of the, I keep going to fruit and veggie now. What was it called before?
CAC.
Canadian Horticultural Council. Great, thanks. It's amazing how quickly we forget it. So now, so the Hort Council are now the Fruit and Veggie Growers Association in terms of their umbrella projects across Canada. And yeah. Now, potato early dying, it's a disease mainly caused by a fungus, verticillium, and the fungus plugs up the water conducting vessels up the stem of the potato plant. Okay? And basically it chokes off the plant in terms of water, and then it can't cool its leaves, can't transport nutrients, it starts to wilt at the leaves, and eventually that can extend throughout the whole plant and die. And it tends to happen later in the growing season because then the choking occurs more then, okay? And it's really the plant chokes itself. It's kind of like getting an infection and taking a belt and tightening it to kind of segregate the infection area so it doesn't kill that area so it doesn't move up. Okay, that's what the plant does, really. Okay? And but obviously that's going to be bad for yield, right? It's going to rob yield. Now there's a second partner, and particularly here, it's quite involved, and that is the root lesion nematode. I'll show you a picture in a moment. A little roundworm in soil. That when these two organisms are together in the soil, so the verticillium and the root lesion nematode, they interact and cause the plant to die a lot sooner, or have greater symptoms of disease and greater yield loss. Okay? And the yield loss is because the plant dies early and you don't have the bulking, particularly late tuber bulking, so you get smaller potatoes. Now notice we have some names here. I wish it was simple and I could just say verticillium in soil, but nah, there's about 3 or 4 species in the soil. And we'll talk a bit more about those. You notice the one I have in bold here, it's going to be figure in big time in this presentation. And for the root lesion nematode, there's also a few species as well. And it's this one species that we'll be particularly talking about or highlighting. Okay. This is the nematode. Probably have all seen nematodes by now in terms of under microscopes or images. Beautiful critter. It's a plant feeding one because it has the stylet here, the spear. The spear will extend out of the head area and that pokes into the root. And then it can suck plant juices, I call them plant juices, into the intestinal area here. And this is the root lesion nematode. How big is this little guy? It's about 1/4 of a millimeter in length. Can you see it with your eyes? Not really. In liquid, you might see a little fleck in there, like almost like there's dust or something in the water, floaty, maybe.
And they go right into the roots too, right?
They go right in the roots, yeah. So this little spear here, we'll call it stylet is the technical term, it will repeatedly go like that, break through the outer surface of the root, or we call the epidermis, and it kind of creates a little, I'm not sure, it's like taking a drill and a piece of plywood and start drilling, and then push through eventually, right? And then break through and then keep poking and get between the cells. Then at some point it kind of gets closer to the center of the root where the vascular system is. That's where the food's going to be moving up, and it'll start feeding and sucking up the juices or the guts of the root cells. Okay? It's pretty awesome in terms of the life cycle. This is the fungus. This is the fungus inside your potato stems. If you slice the potato stem in late August, September, you could see little pepper-like grains in the pith or the center of the stem. Okay, not on the surface of the stem or the outside of the stem. Okay, and inside. This is in the laboratory, grown in cultures and seen underneath the microscope. So one of the key things that we wanted to achieve in the project was to determine what species of the verticillium and the root lesion are present, okay, in PEI. But not only PEI, New Brunswick, Ontario, Manitoba, and so forth. Okay? And we'll show you that. So, this is, so samples were collected across the different provinces and sent over to the analytical lab, State of New Brunswick, the CS lab, and there looked at, particularly for 2 species of verticillium, Verticillium dallii and Verticillium alboatrum. Which of these 2 species is more prominent across Canada?
Dallii, for sure.
Dallii, yeah. Now how about PEI?
Very good.
Yeah, 0 actually. Now, so Dallii. Now, previous to this work, I would have told you from the literature, from the research that had been conducted in the past, it would have been this critter present in PEI. Okay? That research would have been done well over 25 years ago. And the methods have changed. Our ability to identify the fungus, which is different species, has changed. It's improved, okay, because of molecular techniques, you know, the CSI, DNA stuff, and finding out who the killer is and all that kind of stuff, right? So we can apply that now. So it's actually the Dallii. And this really does make sense, and it's consistent across Canada. So that's the beast there that we have to worry about, okay? Now, if we look at the frequency, or in other words, in the samples or fields, what's the, basically 80 out of 100 fields, if we had 100 fields sampled, would have been, have the verticillium dahlia present. Okay? In other words, most of your fields will have the fungus.
And we got some zeros back from the lab, but I'm almost certain they're not zeros.
Yeah,
Because
so-
we didn't really sample any fields that didn't have potatoes in rotation. So.
Okay. So, you know, it was pretty safe to say you grow potatoes, you're going to have the dahlia present, okay? And continuing on that work done in Fredericton with samples, you can see that there is some variation in numbers, so higher amount of the fungus on the vertical here, and there's 30 fields in 3 different years. And you can see the majority, 2/3 of them are fairly decent. If you look at, if we say 200's a decent amount enough. We still don't know exactly what's going on with 2021, but if you look at that other species, not much going on, okay? And so it's everywhere. So how about determining the levels of this fungus? Okay. You have 2 options to grab soil samples and submit those to a laboratory. One of the times that you can sample and send off a sample for knowing the level of the fungus so that you can make some decision about your cropping. What would be the ideal 2 times?
Spring and fall.
Spring and fall, yeah. So that's exactly what was done here, looking at samples that were taken in the spring and in the fall. Same field, year before potatoes. Well, the spring before potatoes, and then the fall previous, year before. And you can see the green are the fall samples, and they're always, you know, not always, but they're higher in the fall, the sample numbers, okay, for the fungus. And this is the relationship of in the fall versus the spring. If they were equal in terms of the numbers, or equal, it didn't make a difference if you sampled fall or spring. This black line here would be like that, okay? So the numbers are, since it's like that, it means actually the numbers are higher in the fall. So what do you think happened between the fall and the springtime to drive the numbers down? Winter, yeah.
Oh.
So we always have a drop in, usually we have a drop in organisms between fall and spring. Okay, now this is, keep in mind, this is the year, this is before potatoes, okay? We've done work in Manitoba after potato, and it's opposite. And it's because the residues break down more in this to the spring and release the fungus. So actually the numbers can go up the year after potato. But you don't really care about sampling the year after potato. You want to sample before potato so that you can make a management decision and say, ooh, I have a lot of levels of this pathogen. Maybe I think about changing on my rotation. Maybe I should think about changing my variety to a more resistant variety. So what now about the levels of the fungus? So Ryan and Daou categorized the levels of the fungus to high and low levels, and that partly based on some previous work in terms of saying how much of the fungus caused disease. And so here you can see the difference, quite a big difference in the amount of DNA. I'm just looking at the spring samples here. I don't know what that is, 6 times more, 7 times more of the high samples than the low samples, okay? At the same time, we also have more of the nematode, the Rhodesian nematode, so it's both culprits are higher, okay? And what's the impact on this, on yield? So there's in metric tons here per hectare. But you can see here, there's four metric tons reduction with the high pathogen.
3,600 weight.
3,600. But what's the difference though?
What, 12%? Something like
Yeah, it's 11% there. So what'd you say, 36?
that?
So we're looking at 4 bags, 400.
No, it's 3,600 weight is the difference.
Okay.
It's 4 metric tons.
Yeah.
It's 3,600.
How many bags? How many hundredweight?
36.
36.
36. Oh, sorry. Here I am saying, okay, I'm screwed up.
We're all good.
36 reduction. The following year, again, the same thing. High for both the vert and the root lesion. And it's even more of a reduction. Do your magic.
54.
Okay. And then in the third year, which was last year,
72.
even more. So it gives you an idea, even here, these are across several different fields, right? But it does tell you about the severity or the so what about this disease for you, right? So the max was 72, you said, right? That's 7,200.
And even in a good year, in theory, like a decent year, we only had one of these fields in Kings County, so, of the 7 in 2022, and it was still a decent yield. It was a good field, but like even the fields with like good growing conditions and look good all along, you could still detect that yield reduction from the high vert and nematode fields versus the others.
Yeah.
So.
So it is a big deal. It's a massive, massive deal.
And these are all russet Burbanks, just so you know. So we're comparing apples to apples.
Let's dive a little bit more into this pathogen, the Verticillium dallii. So Benjamin Tanya at Canada and Quebec looked at the DNA in more detail of the dallii, and they're describing two family offshoots, lineage 1 and lineage 2. In the east, it's lineage 2 in the dark that make up the majority of the fungus isolates, individuals. And in the west, where I come from, it's lineage 1. So it's like one part of the family moved west and one part of the family moved east, you know, and once in a while they make a phone call to each other and they get a little bit of mixing, okay? And so that's pretty cool in terms of, you know, as a research scientist, we love that kind of stuff, right? Geographical separation, right? But you want to know, is my guy worse than mine? Right? So and this is the beginning of looking at that. We got to dive into it more deeper. This is more disease in the greenhouse. Okay, it's called area under the disease progress curve. As the number goes up, that means you get more symptoms of wilting and dying of the potted russet Burbank. Your family lineage is worse.
Isn't that so? Doesn't the bottom it says 1 and 2? So aren't we mostly 2?
Oh, hang on a second.
So we would be the second one, right?
Oh, you're right. I keep going by the dark up here.
No, I think it's by the
Yeah.
so ours is a little bit less.
Yours less. You're in good shape. I'm in the bad shape. Oh no. Oh yeah, we'll have to change that from the slides. You're right. Lineage 1, so should be the gray, light gray, and that should be dark. You're actually better off. Well, that sucks. That pisses me off.
You thought you had one up on us, Mario?
Yeah.
Yeah.
Damn. I'm jealous. So we have to look more into this, especially in field trials. Let's go to the worm. And we said about there's different species of the worm. And the worm and verticillium interact to cause disease. We don't exactly know how they interact. It used to be thought that the worm, through its poking action and getting into the root, allowed the fungus to follow through the holes created. We actually don't think that's the case. Right? We actually think it's that the worm, when it's present in the root, weakens the plant, and the plant cannot protect itself against the fungus as well. Okay? It's weakened. Right? Think of it as its immune system is being weakened by the presence of the nematode. Okay? That's what a few lines of research in the past have indicated. So we wanted to look at the different species of the root lesion nematode. There's 3 species looked at here: penetrans, neglectus, and crenatus. And these are found, all 3 species are found here. And with and without the verticillium. Where the number is higher, that means there's been more reproduction, more success of the nematode species to reproduce and grow and feed on the Burbank. So the gray is no verticillium, and here crenatus is actually doing better without verticillium. Not, the boxes overlap each other, so probably not statistical difference. For Pratylanchus neglectus of another species, same thing. In the presence of the verticillium, it's not happy. The nematode, that species, drops. But look at the penetrans. It's the reverse. It likes to be with the fungus. It grows more, reproduces more. Okay, this backs up previous research that has really shown this to be the case between the Pratylanchus penetrans and the Verticillium dahlia. They're synergistic. They cause more disease. Research in other nematode species, other root lesion species, really hasn't been done. So it's but this is a good news story. Okay, this is same old bad news. Okay, it just reinforces it. Okay. This is Benjamin here at Canada, researcher in Quebec, and he's done all this detailed nematology work. Okay. And here's an example of the different species. There's 4 species here. There's probably 1 or 2 other ones that would make just very tiny slivers you couldn't see in terms of their proportions found in the different provinces. We take PEI here of 400 nematodes examined, individuals. The vast majority, a good, 85% were this crenatus. And then red is the one that's synergistic with the verticillium. Cause more disease, the penetrans. We can look at where the red is. New Brunswick, Ontario, PEI, Quebec. The really bad nematode that's synergistic with Verticillium dahlia is in central or eastern Canada, not in western Canada. Okay. Manitoba and Alberta is an absence. We have a lot more data from Manitoba from my lab. It backs up. We haven't been able to, in 20 years, I've not been able to find the red nematode. Okay. That's good news for us. What does this mean for you? When you're looking at potato early dying control, you're particularly dealing with the 2 critters, okay? The penetrans and the Verticillium dahlia, and where the penetrans is in a field with, and well, you're gonna have verticillium dallii in all your fields, but not all of you will have penetrans. Those of you that have penetrans, you're gonna have to be more wary and careful about potato root dying and get on that nematode control, okay? For those of you that do not have the penetrans, it's not that you take it easy and don't worry. You're still gonna have the fungus, but the bad nematode, the worst nematode, you won't have, so you're gonna have to keep the other nematodes in check, okay? You're gonna have to make sure that they don't get too high, okay?
So if we do a nematode sample now, does it take all the different species into account?
No, not yet. That's an outcome of this project, is that, or as a recommendation, is that we need to figure a way to incentivize or provide the tools to commercial laboratory services to provide that. So through this project, we're able to get the ACS lab to provide that service for the fungus. Now we have to work on for the paramycus. Okay.
And before now, we didn't really know that penetrans were that much worse than the rest of the root lesion nematodes. So, like, we know that they're way worse than the rest. The rest are more or less benign actors. And now we need to kind of get those, okay, if you got 5,000 nematodes in your field, how many of them are actually penetrans?
Yeah.
So,
But I think, I don't want to say that, like, if you come back with if you send off a sample to a lab now, and they come back and say you have 10,000 root lesion nematodes, I would say, whoa, that's a lot. They might be the gray, but that's still a concern, because there's a lot of nematodes to swamp your root system, right? Now, if it came back and said, oh, you have 300 root lesion, okay? Then I would say, hang on a second here. We need to know what species that is. Because if you have, in that 300, if you have the red one, then you have a nematode problem, because it's going to interact with your verticillium. But if that report came back 300 of the gray root lesion, I would say, you can breathe a little bit here, okay? Let's just work on making sure those numbers don't get up to 5,000, 10,000, okay? So we need to work on this to moving forward to give you some of those tools so you can make some decisions, okay? I just don't like it when the results come back from a commercial lab and it says root lesion nematode or verticillium. You know, it's not super informative for you to make decisions, management decisions, okay? All right. Now with the nematode, let's just move on. This is more data from PI, but it's just telling you the different levels. Some are astronomical, okay? If we use 5,000 for the nematode, and that would be for any species, like just in general, the whole cluster of all the root lesion nematode. You know, you can say, again, about half to 2/3 of the samples are above that level. So it's a concern. Because the nematode alone and high levels in soil can cause damage to your plant. Okay? We don't technically call it early dying, but it's going to stun your plants, it's going to rob yield.
they could do like root pruning, right?
It could be root pruning and root damage, but throughout the whole root system, like take a length of root, it's going to be, throughout that, it's going to be damaging in different sections. It's called a root lesion, because if the roots are big enough, you can see a brown, dark browning area. Sometimes it's sunken a little bit, and we call it a lesion. Right? It's a damaged portion. So, now this is a different story from my area. We don't have the penetrans, but we also have a lot lower counts of the root lesion. So we can breathe a lot easier, right? We deal with just verticillium dallii. as our culprit. But here, you have to do a little bit of sleuthing to find out that if you have both of the real bad characters, okay? And then you can also have an issue if you don't have the real bad root lesion nematode. If the levels are extremely high, you need to be worried. Beautiful shot of Ryan here, very happy with his potato hat.
With a field of dead potatoes behind me.
So here, looking at the levels of the nematode in general, and the Verticillium dallii, the specific fungus, following the growth of different crops. Okay? So in other words, think about it, these crops would be before your potato here. Okay? And Ryan went out and sampled saying, hey, are there certain crops that are promoting these critters? Okay? Or reducing them. Let's look at the verticillium in the light blue. The biggest number in terms of the presence was with after red clover. Soybean pea, and Sudan grass. Well, red clover is it's not a great find, right? Because many of you probably use red clover as a rotation crop and a plow down or something like that, right? Soybean peas, another legume, as a rotation crop. Sudan grass, we're a little bit surprised by because in literature that's thought of as a suppressant of verticillium, okay? But mind you, this was only 1 year growth, and as Ryan has pointed out, these growers were probably knew, based on talking with Ryan and him showing results, that they had high levels of verticillium, and probably tried the Sudan grass to suppress the levels, okay? So they may be already high to begin with. What's on the low side? Alfalfa, cereal grains, mixed hay. So those are good rotation crops for verticillium.
Mario, a question I get a lot from growers is around verticillium resistant alfalfa, and is that going to help me for potatoes? And I always say, and I want to make sure that I'm right, that the verticillium resistance, the resistance in the verticillium resistance alfalfa is around other species of verticillium that are attack alfalfa, and not really going to help you with Verticillium dallii. Is that correct?
Yes,
Yeah.
generally. So you may you will hear, if you go to Seed Guide and stuff like that, about alfalfa and susceptibility to verticillium. It's different species. There's alboatrum, that other one, and then there's also Verticillium alfalfae. Pronouncing that a little bit wrong, but it's a different species.
Just want to make sure I wasn't telling lies.
In that case, you're not lying. Other cases, you probably are
Yep.
lying.
Sure.
That's good. Now in the blue, we have the root lesion nematode, okay? And let's look at the good side of things. Below the black line, we're putting 5,000 here. Let's say we don't know what species it is, just say in terms of, we want to stay below 5,000 in general for root lesion, okay? Regardless of the species. Pearl millet is dropping below that line. Radish, is it tillage radish?
Most of it was tillage radish. Some of it was oilseed radish, but
Oh yeah, okay. So radishes, below the line, and also cereal grains, and that makes sense from the literature, previous research. Pearl millet is thought of as a suppressive crop to the root lesion nematode. It seems to be working there, but also the radish, which is cool, and the cereal grains. So that's that makes sense. Now, what's the worst? Red clover.
Do those nematode count numbers include beneficial nematodes?
No, just the root lesion.
we do get, when you get your nematode counts from PQI, they do split them into different fractions.
They
So
can
there's a
accept.
root lesion, there's pin, there's stunt, there's root knot nematodes, then they have a big one that just says other, and that's probably everything, is it just in that other?
Yeah, most of the others are benign or beneficial. Yeah. And there's, what depends, in our agricultural, on our fields, sometimes the plant parasitic nematodes can outnumber the beneficials. But the red clover's popping up there, soybean peas are popping up there, encouraging. The mixed hay, which probably has some legumes in it, is up there. And also the alfalfa, or the root lesion nematode. Okay? So this gives you a little bit more complexity. Okay, now I wish it wasn't this complex, because now you can see the two culprits for the disease are responding differently to rotation crops. Right? So it's not simple saying, hey, just use pearl millet. Well, that's going to work on your nematode, not so much on your fungus. Okay.
The one that surprised me a lot, and I don't know what your perspective on it, Mario, is like generally I wasn't surprised with the grain. So like wheat and barley. Usually after that we don't see big numbers of nematodes because it don't seem to be an issue, and they definitely aren't multiplying vert. So the vert's going down in those years a little bit too, just through attrition. But the one that surprised me is ryegrass, and consistently in multiple trials, even outside of this, we see reasonably high levels of root lesion nematodes after ryegrass.
Okay.
And is that because like ryegrass has such a big root system, or like fine roots, or what's the deal there?
Good question. Now I think I can tell you this probably. When nematodes go up after rye, it's not going to be penetrant. It's going to be the other species.
Okay.
Okay. Pratylenchus, the name of the genus. This is do a little bit of Greek on you. Okay. Praty means of the meadow. Of the meadow. Lenchus is bearer of a spear. So you saw a spear. And guess where the first, 300 years ago, where they found Pratylenchus was in pastures and meadows in Europe. Okay? And so it's not like it doesn't like grasses, but obviously a meadow would have the mixture of clovers or legumes and grasses and stuff like that. So and I know the other species like Neglectus that we have can thrive on cereals and so forth and grasses. Okay, so I think if we were to do a deep dive in the species following ryegrass, I'd be surprised if it was penetrant. Okay, so we have more work to do. It's another, I think, to line
Add to the list.
Yeah. Okay, let's breeze through this really fast. So we're also screening other pathogens in soils, and this is just really the beginning of the idea that you can take a soil test, submit it to a lab, and get a pathogen screen of potential potato pathogens in your soil. Okay, so let's just take a look at this, and let's look at the relative difference between PI and the other provinces. So Rhizoctonia, Rhizoc, and PI, the black is the key thing to look at here, this black line, and it's a, bye guys, thanks. It's down there, baseline basically. Quebec has higher levels where the line is, especially under the muck organic soils. Okay, New Brunswick higher, Manitoba higher. You're fairly low. There's a few samples, few fields that are high, so some outliers there. We look at black dot or Colototricum. PEI is on the lower side compared to all the other provinces. That's a good thing. In my neck of the woods, we're concerned about black dot. And if wherever early dying field we also have black dot. So we think it's a complex of Verticillium dallii and black dot causing our early dying. And we're on the high end here. Okay? So
Is it more prevalent in a dry year or anything?
the black dot? No. Most farmers would say black dot is there always. Yeah.
No, I called Ryan a couple times last year on the field that we were having issues with, and I think that's what it came back to.
Is that right, Ryan?
You did, I don't think you actually
We
got a
didn't-
chance to look at it.
No, but we were talking about it, and you were thinking that that might be an issue.
So if you can see those pepper grains on the outside of the stem when it dies, it's an indication of black dot. The pepper grains are in the inside of the stem, it's not black dot, it's verticillium. And then also, it's a little difficult, like you get the lower leaves that start to die with the black dot. Whereas with verticillium, it's the upper leaves that start to wilt. So the symptomology is quite different. And
First
if
time
you-
I seen it, when it strips almost in the field.
Pardon?
They died off in almost like strips.
Like that?
Yeah. We had 2 fields like that of Russell Burbank.
So some strips died early, some didn't?
Yeah.
Could it be nitrogen? Like a variability in the nitrogen application?
There's no pattern to it that way, I don't
Oh.
think.
Oh, interesting. Could
And
it
then we
be?
used a product from BASF and seemed to stop it.
Oh, yeah?
I figured the field was gonna be dead, totally.
Was it
dead.
early? Early blight?
No, but it was I can't even remember the name of the product, man.
BSF, I'm not sure. Serenade, somebody say serenade?
Chris?
No.
Powdery scab. Sometimes can be confused with common scab. Sometimes in this silver scurf. And you folks are on the bottom. That's a good thing. I'm on the high end. That's not a good thing. And we're concerned about powdery scab. Pythium causes pythium leak in storage. You're on the bottom end, which is a good thing. So actually, these other pathogens are doing pretty good. That's pretty awesome.
Comes from a dependence on PEI grown seed.
Of course he's gonna say that. Because all right, I'm going to basically, do you want to continue with these other products here? But they don't really help, they're not really,
A lot of the people here are used to, like, they've seen some of the work that we've done on, like, vellum prime and some of these other things.
Yeah.
So, and we don't fumigate here, so that's not really.
yeah, so we're gonna skip that. Basically, I'm saying, we tried everything under the sun that possibly we could throw at the plants in terms of chemical control products. There's nothing really super well-defined for verticillium, right? And we do get yield boosts. This is Khalil's work in New Brunswick. He's getting like 25% yield boost when you start stacking these products. And we as well seen, not as well as defined or improvement as Khalil found, 5–10% stacking of these products. We do find fumigation in like 1 in 5 fields really bumps up the yield, and particularly in fields that have low yield to begin with. You know they're a tired field. They're below 300, more around 250. We know if we fumigate, we got a yield, massive yield response, 80, 100 bags. Okay, and it's verticillium and early dying. That's the problem. So, and I think the similar situation is here. In that you got, those are the most responsive fields that you have. That's going to be responsive to any kind of good management practices. And Ryan will mention a few of these when he gives his talk. Okay. So there are lots of people to thank, and in terms of particularly you for helping to fund this project, and Ryan for the activities in PI. Okay. And we'll have any questions on discussion on the verticillium and early dying before we go to the nitrogen?